Hello friends, welcome back to another video from Shomu's Biology. In this video, we are going to talk about how to answer complex and difficult part C questions from cell biology. Okay, so we are looking at different units of CSIR NET life science examinations, and this is unit 2 cell biology. The type of question that they will ask will be different. Now, most of the time you will see that students get confused when they see some of the part C questions. Although they have the knowledge, they read those topics completely, but still when they present the same topic with different formation of a question in part C, it sounds and it looks difficult. So we are going to try to see how exactly we can break those complex difficult part C question into smaller fragments in easier steps so that we can answer this question better way and faster. So let's look at it. The very first question about the cell biology is here. In eukaryotic chromatin, 13 nanometer fiber, which is also known as solenoid, can open up to give rise to two kinds of chromatin. In type 1 type, that is type A, the promoter of the gene within the open chromatin is occupied by a nucleosome, whereas in type B, the promoter is occupied by histone H1. The following possibilities are suggested. So basically, once you read this question, it's a mouthful of question. You need to read a lot, and also you need to go through these are not options, these are statements. So this is a perfectly formed question for part C in CSIR NET life science. This is known as which of the following is correct type of question, and 70% of part C questions are composed of this kind of questions, which will kill your time, which will allow you to read a lot of things, confuse you, and ultimately you made mistake. So what you need to do is that you read this question. You know one thing, that this question is asked from nucleosome model of and how exactly the DNA is wrapped around histone. So basically this question is from that side. So if you have not prepared how exactly DNA is wrapped around histone, right, histone H3, H4, H1B, H1A, as well as this other histones, H2A, H2B, H3, H4 and H1 histone, all these different kinds of histone. If you don't have any knowledge about that, you cannot answer this question. So you can leave it right now by reading it for 30 seconds. But if you know that, you can answer this. Now try to understand the very question. The question is asked from a simple place, that this is DNA and this is histone. It's okay. So let me change the color for the histone here. Okay, let me draw the histone with blue color. This is histone and red is the DNA. So DNA is wrapped around histone. So basically the histones the DNA is wrapped around is H2A, H2B, H3 and H4. These are the four type of histone the DNA is wrapped around. But H1 histone has a totally separate role. H1 histone basically binds specific region between the DNA and the rest of the histone octamer to pull the histone close to the DNA and to make it more compact, to make the structure more compact. That is the role of H1 histone. Okay, this is what you need to know. You need to know the structure in order to answer this question. Now it says that there are two promoters that let's say the cell try to access for transcription, and the promoter type A is actually present somewhere here. So let me change the color. This is, let's say, the type A, and this is where type B. It says the first promoter, type A, is actually blocked by the histone octamer, that is by the nucleosome, right? By nucleosome means the histone octamer, and the type B is actually blocked by H1 histone. Okay, so what will happen? The gene in A repressed, the gene in B repressed, the gene in A active, the gene in B active. Now what will happen? At this present moment, if they are blocked by either nucleosome, that is HA, H1A, H1H, 2B, okay, H3, H4, H2A, H2B, sorry, H2A, H2B, H3, H4, not, it should be H2B, not H1A. H2B, H2B, H2A, H2B, H3 and H4. All these histones, they are going to be present as a nucleosome. So if they block, obviously we need to release the block, otherwise we cannot access the promoter. Similarly, in H1 histone, if H1 blocks the promoter, again we need to release the H1 histone in order to access it. So basically at the present moment, it doesn't matter whether H2A, H2B, H3, H4 histones block the promoter or H1 histone blocks the promoter. In either way, if the promoter is occupied, if it is blocked, then the promoter cannot be accessed by the RNA polymerase. So the expression of the gene will be blocked in both the situations. So basically the gene A will be repressed, the gene B will be repressed. So A and B both are standing true. So let's find out option with both A and B, and that is option B. So in our case here, what we can say is that option B is the correct answer. It must be the correct answer here. Okay, so got the idea. This question is asked from a very simple point of view. If I asked you a question that, let's say, don't look at this question, but let me ask you a simple question that DNA is wrapped around histone, okay, and if a promoter is present and blocked by that histone, then what will happen? Can we access the promoter? You would say no, there is no chance. If I tell you that yes, there is a region of the promoter, but it is covered by H1 histone, okay, without removing H1 histone, can we access the promoter and express the gene? You would have said no, there is no chance, because they are blocked. We need to remove them in order to use the promoter, access the promoter. You could have said it very clearly, right? By knowing a very simple fact that if they are blocked by these proteins, they cannot be accessed. But when they form a question like this and telling that there is type A promoter, type B promoter, sometimes they can also give you names for this promoter. Instead of type A, type B, they can give you different gene names. Okay, the gene names can also be written. In that case, let's say MCD8, MCD10, like that, for example. So they give you that kind of name. So what will do? What you will do? You'll get confused that what is this? I don't know that. So this is how they make the questions complicated. Ultimately, it is so simple as this picture that we shared. Okay. Now let's look at the next question. Which of the following statements regarding membrane transport is false? So basically this question is from membrane transport. If you know membrane transport, you can attain this. And basically all of you must know membrane transport by now. Polar and charged solutes. So see, this is a question which will take some time, because this question, these are easier questions basically. The questions which are combination type, this type, are complicated. That's why they compose most of the questions with this one. But this kind of questions which are direct question where the options are lengthier are easy to answer. Polar and charged solutes will not cross cell membranes effectively without specific protein carriers. So true or false? See, the question is you need to find out the false statement. So basically among these 4 statements, 3 of them are true, 1 is false. You need to find out the false statement. Polar and charged solutes will not cross the cell membrane effectively without specific protein carriers. Yes, true. Because charged molecules require channel proteins to carry. Each protein carrier will only bind and transport one or a few similar type of solute. Not one, but yeah, very few similar, it's true. Sugars such as glucose are always transported by active transport rather than facilitated diffusion carrier. This is a false statement, because we have seen multiple examples where the sugar is carried through the membrane in a facilitated diffusion manner. Sugar is also carried through membrane in a indirect way of transport. So sugar not always transported by active transport. Yeah, secondary active transport can be involved, and also facilitated diffusion of sugar is also possible. So this one is a wrong statement. So we found our false statement. Let's read the last one also. Ions are typically transported by special proteins that form membrane channels. Yes. So that's also true. So we know among this 4 statement, C was wrong. So basically, if you read this question, this was a big question, formed a part C question. Now I can ask you a simple question like that. Do sugar move across membrane via facilitated diffusion? Okay, this is a single line question. Do sugar or can sugar move across the membrane via facilitated diffusion or not? Single number question. If you say yes, it can move. That is basically the question. See how exactly the question is formed. This is the question. And what CSIR did, they convert it into this question. So this is the actual framework of the question. So when you read a question, you need to break it down, shred it out to find out the simple single nature of the question. Let's look at the third one. Mitotic cyclin CDK activity peaks in M phase. This is because. So basically single question. why M cycline CDK complex is highest, present in highest concentration at M phase. So basically why and how cycline CDK for M phase increases in M phase. We know that there are M phase cycline, there are G1S phase cycline. So basically G1S phase cycline will increase in the G1S phase, that transition. M phase cycline will increase in concentration during M phase. Why does that happen? Basically that is the question. Okay, but it is again formed into another this kind of question. Why? There are different statements provided. Mitotic cycline is synthesized only in M phase. True or false? Mitotic cycline is synthesized only in M phase. No. See, the cyclines are there. Okay, CDK and cycline partners are there. It's not likely this is only produced in M phase. Threshold level of mitotic cycline accumulates only in late G2 phase. Cycline subunit is activated by phosphorylation only in M phase. The kinase subunit is activated by dephosphorylation only in M phase. So basically they are saying that whether the M phase cycline is present only in M phase or at late G2 phase, that's why the cycline CDK activity increases, or the cycline subunit is activated by phosphorylation, or the kinase subunit is activated by dephosphorylation. That's what you need to know. So alongside what you need to know, basically the CDK and cycline combination, cycline M and CDK combination. So basically cycline M and CDK combination is very important. CDK is the actual enzyme. That's what you need to know. The basic interaction between cycline and CDK. If you don't know that, you cannot answer this question. You need to know that. You need to know that cycline and CDK partner is very important. CDK is basically the enzyme. So any kind of phosphorylation, dephosphorylation, all these things will be done for the CDK and not for the cycline. So of course C is not true. Cycline is not modified in that way. CDK is the enzyme that is modified. The kinase is the enzyme that is modified. Where cycline CDK partner is always there. After the work is done, the cycline is destroyed. If the cycline M will be destroyed after the work is done in the M phase, then the CDK, which is cycline dependent kinase, without cycline cannot function. So basically cycline concentration will be there, slowly accommodate from the G2, late G2 to the M phase. That is kind of a true idea. Yeah, there. So it's not likely the cycline is only synthesized in the M phase. No, but slowly accumulate through the cell cycle till the M phase. And when the cycline concentration is higher, CDK associate with cycline partner, that is cycline M, and then CDK is dephosphorylated, and as a result of the dephosphorylation of inhibitory phosphate that was added to the CDK cycline complex becomes active, and that's why D is the correct answer in this case. Okay, so basically A, B, C, these are not true. D is the true statement here. Because we know that dephosphorylation of CDK makes it active, CDK along with cycline M can function. Once the function is done, then the cycline M will be destroyed, okay, by the anaphase promoting factor anaphase promoting factor APF at the end of the mitotic phase, and that initiates the process of anaphase. Or metaphase is completed, now anaphase will begin. So this is the whole scenario. So basically you need to know this idea of cycline CDK interaction. You need to know which is activated, which is the enzyme that can be activated by phosphorylation or dephosphorylation. Then only you can answer this question, otherwise you cannot answer the question. It takes some time, but you need to exactly understand what is the question. The question is why M phase cycline CDK activated and work in the M phase only. Okay, let's move to the next question. During cell cycle progression from G1 to S, cycline D CDK 4 phosphorylates retinoblastoma protein and reduces its affinity towards E2F. E2F dissociates from RB, activates the S phase gene expression. Okay, overexpression of protein A arrests G1S progression. Which of the following statement is true about this protein A? So this, it can be protein A, B, C, D, X, Y, Z, whatever it is. So basically when you are reading this question, you need to know one fact, that this question is asked from retinoblastoma sensitivity protein, that is PRB, and E2F combination, and also G1S progression. So cell cycle progression from G1 to S, and also at this moment, that is PRB associated to E2F. And remember, E2F acts as a transcription factor for several downstream genes that helps the cell progression or cell cycle progression. So the thing is, if retinoblastoma is phosphorylated then what happens is that it releases E2F, and E2F is now is free. E2F allows the progression of G1 to S. That's the normal situation. You need to understand this interaction between E2F and retinoblastoma PRB protein. If you don't know that, leave this question. But if you know that, if you recall that, then answer it. So what happens in very simple terms? PRB phosphorylated, it releases E2F. E2F causes G1S transition. Now we are talking about a protein, a hypothetical protein here. Okay, let me take the color. A hypothetical protein, and that is what we have. Let's move. Yeah, the hypothetical protein is A. What A does is that arrests G1S progression. So this protein basically preventing the G1S transition. Okay. So if A is preventing the G1S transition, that means A is not allowing E2F, activity of E2F. So that means A will not allow the phosphorylation of PRB. So PRB will remain associated with E2F. So no E2F, no progression of G1 to S, right? Isn't it? So following statement which are true: A inhibits retinoblastoma E2F interaction, A inhibits CDK4 activity, A phosphorylates E2F, A degrades retinoblastoma. Which among the four is correct? Think about it. A inhibits retinoblastoma, like RB-E2F interaction. If the interaction is inhibited, then the progression will be easier. So definitely A is not true. Try to understand. If A inhibits retinoblastoma E2F interaction, then E2F can be free, right? So this interaction is not inhibited. A inhibits CDK4 activity. During cell cycle progression, cyclin D CDK4 phosphorylates retinoblastoma. So basically this phosphorylation is done by cyclin 4 and CDK4 and cyclin D. A inhibits CDK4. So what happens if A inhibits CDK4? Then CDK4 will not phosphorylate PRB. Then E2F will not be free. Then there won't be any progression. Got it? Try to understand. If this A protein inhibits CDK4 activity, then there is no phosphorylation of PRB, because in the question told the CDK4 and cyclin D phosphorylates PRB, and we know phosphorylation of PRB means E2F is active, means G1 is progression. So CDK4 is inhibited by A, good. As a result, no dissociation of E2F. No dissociation means no conversion or activation of further genes that will allow the cell to move from G1 to S. So basically B is the correct statement we can choose, that yes, A, this hypothetical gene A inhibits CDK4 activity, and as it inhibits the CDK4 activity, no dissociation of E2F, no conversion or no movement of cell from G1 to S phase. So that's the answer. A phosphorylates E2F? Definitely not, because phosphorylation of E2F is nothing to be considered here. A degrades retinoblastoma? No, because degradation of retinoblastoma protein or E2F will not, it will always help the conversion of G1 phase to the S phase, but we are looking at inhibition of G1 to S phase, and that's what we can talk about. So option B must be the correct answer in this case. So see, in this particular question you need to visualize the whole mechanism. This is a mechanism based question. You need to visualize the mechanism, how exactly the process takes place in order to answer that. Let's move to the last question. The tetrapeptide KDEL is well known as a retrieval signal of several newly synthesized proteins. This process is mediated through specific receptor KDEL interaction. Any single amino acid change in this tetrapeptide is not allowed. Even a single change is not allowed in terms of binding with its receptor and subsequent retention in specific organelle, whereas secretory proteins are devoid of such tetrapeptide. From this observation indicate the localization of the receptor of this tetrapeptide. So basically this is a whole new nonsense till this moment. The whole new nonsense is provided. That's why I put this question in here. You don't need any of these statements. You don't need all this huge information regarding what is KDEL, what is it. So basically KDEL, KDEL, these are all amino acid single letter codes. Okay, tetrapeptide. This code, this is present, and whenever in the protein if this code is present, the protein is retrieved in the retrograde pathway, that is in the backward from the membrane towards the endoplasmic reticulum. So from Golgi apparatus via vesicles into the endoplasmic reticulum, that mode of transport is actually continued. So any protein with this KDEL signal is retrieved by the cell in the opposite direction. Normally the forward direction is endoplasmic reticulum, the vesicles, Golgi apparatus, vesicles and cell membrane outside of the cell. That is anterograde transport. Retrograde is the reversal, opposite direction. So retrieval pathway is retrograde pathway. We don't need to know this. Only thing you need to know is that where exactly the KDEL associated protein and KDEL receptors are found. So basically, if inside the vesicle we have KDEL receptor, then the KDEL will bind to the KDEL receptor and they are retrieved inside. This is the idea. Okay, so let's imagine this is the cell membrane and this is the Golgi apparatus, and basically this is the endoplasmic reticulum. So ER, Golgi and vesicle. Okay, so this is how interaction works in the retrograde model. So if KDEL receptor is present, it will be inserted inside. So basically KDEL receptor is located, and KDEL receptor interaction located in which of these organelle? Plasma membrane? Of course not. Golgi, endoplasmic reticulum, mitochondria. Of course not mitochondria. Endoplasmic reticulum and Golgi, between the two. Of course here we are talking about Golgi apparatus. Because in endoplasmic reticulum the proteins are produced and synthesized. They don't have KDEL receptors there. The KDEL receptors are present in the vesicles that are coming out of Golgi and inside the Golgi itself, where the proteins with KDEL signal will bind and they are taken inside. So retrograde pathway is involved. Okay, so that is the idea about KDEL receptor. That is the idea about this question. So basically you don't need to read any of this. You need to know that the KDEL receptor is present in the Golgi apparatus and the interaction will work in the Golgi apparatus to answer that. It will take 15 seconds for you to answer it, but CSIR wants you to read all this and make it more confused and waste your time. Okay, so this is how it works. I have already mentioned. Now a culture medium contains two carbon sources. One is preferred carbon source, let's say glucose, and second one is non-preferred carbon source, let's say lactose. Okay, which one below is correct regarding the nature of growth curve of E. coli in this cultured media? The moment you're reading this, you know that this is a diaxic growth problem. Diaxic growth means where the bacteria is provided with two different carbon source. One is readily available one, the easier one, another one is a little complicated one. So the readily available one is glucose and complicated one is the lactose. So of course if you give two types of food, one is readily available and another is complex, obviously the organism will try to eat the readily available one first, then will go for the complex type. So they will first take glucose. So basically the growth curve, what we know is something like this, right? So lag phase, log phase, stationary phase. Okay. Okay. There is some calibration error, I don't know what's going on here. Lag, log and stationary. And now what we can clearly see at this moment is that, so this is for one particular type of carbon source, one particular type of carbon source. Now this carbon source type can be changed. So here it shifts from one carbon source to the another carbon source. So it will take some time. So another lag phase will be present and then there will be log, then there will be stationary. Okay, so this is for the first kind of carbon source, this is the second carbon source, this is glucose, this is lactose. So growth curve will be same as when grown in the presence of only glucose? Definitely not. Growth curve will be same as when grown in the presence of only lactose? No. A lag phase will be observed between two exponential phases, two lag phases will be observed between the two exponential phases. No, only one lag phase. Basically at this moment, at the beginning, it will start taking up the glucose as a carbon source to continue to grow, but once the growth is done, it will be stalled, the stationary phase. After that, the glucose will be depleted and now they need to make the enzymes adjust themselves to produce all the necessary enzymes to utilize lactose as a second carbon source. So when they take some time for adjustment, another lag phase is there. So basically one lag phase will be present between the two exponential phases. So C should be the correct answer in this case. So basically once you read this question, you know it's a diaxic growth problem. You know, if you heard the name, if you know what diaxic growth, you can answer that. Otherwise you must leave it. So this is the type of question that you can see. We have talked about the question, how they make it more complicated, how they make it lengthy, but you need to break it down so that you can answer it fast. So these are the type of question you can expect. If you like this video, please hit the like button, stay tuned and subscribe to this channel because this kind of lectures are coming soon that will help you take perfectly formed decision in the CSIR NET exam hall. So all the very best. Thank you. Bye.